In this module, we will study how the target sites locate for the transcription factor during the cell control. Now, the genes, they are stimulated by activators which bind to enhancer. They are stimulated by activators, and these activators, they bind to the enhancer. Now, many activators have many enhancer targets in a genome and therefore activate many genes. What I said, many activators have many enhancer targets in a genome and therefore activate many genes. Now, these set of genes that tend to be regulated together is sometimes called as a regulon. Regulons are set of genes that tend to be regulated together. And one of the strategy which was employed by Richard Young and colleagues meld two different techniques. There were two different techniques which these guys, they applied. One is chip, that is where chromatin immunoprecipitation and then DNA microarray hybridization on a DNA microarray on a chip. So there are two processes, that is the chromatin immunoprecipitation, and second is the DNA microarray on a DNA microarray chip. Now these two, when they are combined, this technique is called chip technique. Now, the general plan for the method is, which Young and colleagues adopted, to identify the binding site for the activator, that is the GAL4, throughout the yeast genomes. That is, they identified the binding site for the activator GAL4 for the yeast. And in this figure, it is demonstrated that how the genome-wide search for DNA protein interaction in a yeast by chip analysis occurred. And on one side, there are the activators, then the genes which are there, they are just hybridized on a glass chip, and that makes a chip microarray which is analyzed. Now, this method is suitable for yeast genome because the yeast genome has a limited size, and the fact that the yeast genome has been completely sequenced. So the two things is that the yeast genome is a limited size, and the second is the whole genome has been completely sequenced. So this is why this method can be used in the study of the yeast. Now, Peggy and Fernham, another group, they used a microarray containing only CPG island. That was the another technology where CPG islands, they were used. And CPG islands, they are associated with the gene control regions, and they should be highly enriched in the activator binding sequences, which is being sought by this technique. So the CPG island, they are associated with the gene control region, and they are highly enriched in the activator binding sequences, which is being sought by this technique, by the activators. Then there are limitations of chip technique, and it is limited to the sequences which is placed on the chip. One thing is, the limitation is that the sequences which has been placed on that microarray chip, it is limited to that. And second is the hybridized efficiency to the spot on a chip is different for different DNAs. For different DNAs, the hybridization frequency, the hybridization efficiency, that will be different. So some binding sites will be missed because the efficiency of the hybridization for different DNAs is different. So the different DNAs can be missed in this technique. Now another technique or another is the tag sequencing or an alternative technique. And what is done in this technique, that the amplified pieces of DNA, they are precipitated on the chip, and the procedures for this hybridization to a chip are not hybridized to the chip but repeatedly sequenced using one of the new high throughput. That is, in this tag sequences, the hybridization is not used, although the DNA is precipitated to the chip, but they are not hybridized to the chip. Rather, they are repeatedly sequenced using one of the new highly throughput technique which is used for this purpose. So in this module, we have studied the three methods where we can find out that how the sequencing or how the target DNA can be found out in the cell.